Okay friends, so today what we're going to do is I'm going to show you how an animal develops from a zygote into, an organism that has 3 layers of cells, at least in embryological development. Okay, so this is basically another way to say this is how to build an animal. And this is going to describe pretty much every animal in the kingdom Animalia. Okay, so we're going to start with the zygote. And you remember zygote, first diploid cell made up of an egg and a sperm. And what happens to that zygote? It's going to divide mitosis into 2 cells, into 4 cells, 8 cells, so on and so forth. And eventually it's going to turn into, I don't know how many cells this is. Just draw a border around that. And this is a solid ball of cells, which is called a morula. Solid ball of cells. And when I say solid, I mean think like a golf ball is solid, or what's another? A bowling ball is solid. Not like a hollow ball, which is what comes next. So this solid ball of cells is going to grow and develop, and I'm just drawing a cross section through a sphere just to give you a sense that it's supposed to be a circle. So now this is a hollow ball of cells. Like a tennis ball or a basketball or a soccer ball. This hollow ball of cells is called a blastula. And a blastula has a cavity inside of it, which is called a blastocele. Okay, so the blastocele is this cavity inside this hollow ball of cells, which is the blastula. Okay, next thing that happens is your lead breaks a little bit, and you make a rude sound to cover it up. And so the next thing that happens with our hollow ball of cells is it starts to get poked in as the cells are dividing and growing. The next thing they form is a hollow ball of cells with one end poked in. So we still have a blastocele. And we still have an outer layer, but now we've poked in one end and formed what's called a blastopore. Okay, so remember that blastopore, that's going to be important later. And that blastopore, now it's not shaped like a U-shaped thing. Imagine this is 3-dimensional, so imagine like someone took a tennis ball and just kind of poked their finger in one end to make a cavity inside. So there's still the blastocele, which is like the air inside the tennis ball, but like this, the blastopore that get poked in, if you poked your finger in there like in a tennis ball, this would be covered with the orange fuzz, just like in a tennis ball. But it is changed. So something interesting has happened here. One thing that's happened is now we have this blind pocket in here, which is called an archenteron. But we have not just one layer of cells like we had over here in our normal blastula. We've got 2 layers. We've got an inner and an outer layer. Okay, so the inner layer is endoderm, inner skin, and the outer layer is called ectoderm, which means outer skin. So we've got two germ layers now. And the word that we have for that is diploblastic, which means two germ layers. Now, if we were forming one of the group formerly known as radiata, which means if we were forming a ctenophore, or if we were forming a cnidarian, call it the name of the phylum, this is where we'd stop. We would get off here, and because they only have two germ layers, and that is what they develop into mature organisms from. But that's not for you and me and most other animals. So what's for you and me and other animals, I'm going to do this one first down here. So I'm not going to draw all of the layers of the cell. I'm just going to draw our kind of developing gastrula. Oh, sorry, I forgot to mention. This up here, this stage is called the gastrula. So that's a gastrula. Okay? So, gastrula has two germ layers. Next thing that happens is that some of the cells inside the blastocele, but attached to the endoderm, start to form a new layer. And this new layer, it's not endoderm, it's not ectoderm. This, my friends, is mesoderm. So if we've got ectoderm, and we've got endoderm, now we have three germ layers, and we can say that this organism is now triploblastic. Triploblastic, three germ layers, endoderm, ectoderm, and mesoderm. Okay, so let's say that this organism is going to keep growing and developing, and eventually what's going to happen is the archenteron is going to keep developing until it pokes all the way through, and the body plan starts to elongate. Because now that we're not in these radially symmetric organisms up here, we're getting into the bilateria, which is all the rest of the animals. So now we've got the blastopore down here, and the archenteron is poked all the way through, and we've got ectoderm, and we've got endoderm, and now all inside of here, this cavity, the blastocele, is now completely lined with mesoderm. Okay, so here's where I'm going to switch gears and use some color just to make things clear. Okay, so now hopefully this will work. So now endoderm is in blue, and this is very, very common. You'll see this in your textbook and all other places. A lot of other textbooks, they always go with blue for ectoderm, and we'll go with red for mesoderm. And that's usual. And then usually they use they use yellow for endoderm, but I don't have yellow on this pen. So I'm just going to make the endoderm green. So endoderm is green, mesoderm red, and ectoderm blue. Okay, so in this particular type of organism, the blastopore is going to develop into the mouth. And because the mouth develops first, we call these organisms protostomes, which means first the mouth. Okay, so protostomes is going to include all of the Lophotrochozoa, all of the Ecdysozoa. So we have the non-molting protostomes and the molting protostomes and a few other things, a few other like the acoelomates. These are all protostomes. So you might wonder, well, where am I in all of this? And the answer is we also are triploblastic, but we develop a little bit differently. And this is new information from the last time I made a video like this. So here's what happens to you and me and a number of other things, is we develop to be triploblastic, but we still have our gastrula that has ectoderm, and it still has endoderm, but our mesoderm actually arises from a pinching off inside of the archenteron. So over here, remember that mesoderm in the protostomes kind of started on the inside of the blastocele, but here it's getting pinched off from the archenteron. But here's kind of a funny, strange case of convergent evolution, is that our next step, and I'm just going to draw it all in colors over here, looks very similar to what we saw, where we've got ectoderm, and we've got mesoderm, and we've got endoderm. Oh, and I should mention that this, obviously, if this is the mouth, this leads into the gut. So where this pokes through at the other end, that's going to become the anus. Okay. Over here, so where the blastopore started down here, still there. But in these organisms, guess what? The blastopore is going to become the anus. And this opening up here is going to become the mouth. So we still have a complete gut, a one-way gut with a beginning and an end. But instead of the blastopore becoming the mouth like it did over here, the blastopore is going to become the anus. And for that reason, these organisms are called Deuterostomes, which means second the mouth, which I guess sounds a lot nicer than calling them protanuses or protoprocs or something like that. Just drawing attention away from the whole fate of the blastopore in this case. So who are the Deuterostomes? The Deuterostomes are Echinodermata, which are kind of interesting organisms because they usually have radial symmetry, but they have pentamerous radial symmetry, 5-part radial symmetry. So these are things like sea stars and sea urchins and sea cucumbers and some other things. But when they're larval, they're still bilaterally symmetrical. So they retain that, and then they have this secondary gain of radial symmetry. And the other group, which should sound familiar to you, are all the chordates. So the chordates include the vertebrates and some other organisms. It turns out that we are a special case of the chordata. There are a few other groups of chordates that are not vertebrates, but we are in this group down here. So just like that, we've managed to generate all of these animals. I'm not exactly sure what happens to sponges, but so we're just not going to worry about that for now. But you can see we've gone from having a zygote, morula, blastula, gastrula, and then the fate of the gastrula is to become triploblastic in these animals down here. And you can either have the blastopore turning into the mouth or the other opening becoming the mouth and developing a complete gut. Okay?